This is the second video for photosynthesis. So you'll be looking at the leaf epidermal structures, and first things you'll note throughout the epidermis are these banana-shaped cells. And these structures, these are called guard cells. They open and close to allow gases to come in or out. So their definition, this is to regulate gas exchange. These are your guard cells. Inside your guard cells, you might see a nucleus. So you might see a nucleus, and you'll also see little chloroplasts. So we have our, most cells have a nucleus. This cell is no exception. And then we have our chloroplasts. Chloroplasts. This area in between the guard cells, it's not a physical structure, but we still give this a name. This is called a stoma. Don't confuse this with stroma in the chloroplast. This is stoma, which means mouth. Okay, so the plural of stoma is stomata. And the size of this stoma, as you can figure, can increase or decrease based on the situation of the plant. Now, surrounding this, you'll have other typical epidermis cells. These guys might also have a nucleus, but nothing special here. So we'll just call these ordinary epidermal cells. Okay. Okay, now for a leaf cross-section. So we're going to start from one bottom or one end, and here we have this familiar structure. We have our guard cells. And flanking the guard cells on both sides are going to be regular good old epidermis cells. Okay, so guard cells, epidermal cells. A thin layer below this, here's this new structure. We call this a cuticle. And let's take a look at this for a sec. The cuticle is a wax-like substance. So we know that waxes fall into the class of lipids. The general definition of a lipid means, it doesn't mix well with water. So this is, not very soluble in water. Or you can say that these structures are nonpolar or hydrophobic. This tells me that if a water molecule approaches the cuticle, it won't be able to cross. So H2O cannot cross. Not cross, so the final definition of a cuticle is it prevents water loss. Prevents H2O loss. Okay, so guard cells, epidermal cells, and cuticle. Here we have the stoma. Now going into the cell, you will see circular structures, loosely packed. And these structures, we call them spongy. We call this spongy meso, which means middle, as the middle of the leaf. Spongy mesophyll. Those little green dots I drew are chloroplasts. So you can say here that some photosynthesis occurs here. Some photosynthesis. But the fact that they're so not, tightly packed, this allows gas exchange to occur. So notice these air spaces. So we call this an airspace, to allow gas exchange. To allow gas exchange. Okay, so further up, so going even higher up, you'll see a new structure. These guys here are called palisade mesophyll, and they're columns put together, very organized, and they have many chloroplasts per cell. So this is the palisade mesophyll, and they have many chloroplasts. any chloroplast per cell. So you can imagine the primary function of these guys. So the primary function is to conduct photosynthesis. Okay, further past that, we'll have regular old epidermal cells. You won't see any guard cells on this side, but you will have a cuticle on this side. The face with the guard cells, we call this the lower epidermis. So this would be the lower epidermis because the guard cells are here. Lower epidermis, and up here would be the upper epidermis. The upper epidermis. Okay. So last set of structures inside the plant, inside the leaf, you will have something called a vein. You know, veins in the human body can transport things. It has a similar function in the leaf. So here would be a vein in a leaf, or it can be also called a vascular bundle. And inside are two different structures. So you have, thicker circles, which we call xylem, and then you have, thinner circles, which we call phloem. So let me first label this. This sheath of the xylem and phloem, we call this the bundle sheath. Bundle sheath. These set of tubes, we call this xylem, and this we call the phloem. Now your xylem, this is going to obtain, minerals and water from the roots of the plant. From the root of the plant. The phloem, the products that photosynthesis make, one of them is glucose. It's going to take those manufactured organic products and send them away. So we're going to say it's going to send manufactured organic products, one of which is glucose, away from the plant to other parts of the plant. Okay. So here is my leaf cell, and let's label a few things. So cuticle is hard to see, but it would probably be this very thin layer up top. Next we have, typical good old epidermis cells. And on this face, we don't see any guard cells. How do you know that? Is because we have guard cells on this face. So this tells me this is the upper epidermis. So that means close to this side should be the palisade mesophyll. And we do have some palisade mesophyll. Here's a pretty good, let me use a different color. Here is a pretty good set of palisade mesophyll. I'll just say PM for that. Okay, going further down, we have these circular-ish cells, not so tightly packed. This is the spongy mesophyll. Here, these openings right before the guard cells, that is your stoma. And then here are your guard cells. Okay, and last but not least, we have some xylem tissue right there, and surrounding it is phloem. So surrounding it would be phloem. Okay, so for today's lab, we are going to react starch with a leaf, and that's because we know that excess glucose. So if you have, these monomers, if you put them together, which is these are products from photosynthesis. Products from photosynthesis. And let's add, a couple more ... This is going to become starch. So excess glucose is going to become starch, a storage form of glucose. And we know that this is going to react with iodine. So react with iodine. Okay, but now why are we going to use heated ethanol in this experiment? And that's because we need to remove the chlorophyll. Remove the green chlorophyll, or else we won't be able to see the reaction. Remove the green chlorophyll. And if you add heated ethanol, it's going to weaken the cuticle and also the cell walls of these structures. Okay, what that's going to do is this is going to allow iodine to enter the cell. And this is also going to allow chlorophyll to leave. And you'll notice your solution will be green. That's because the chlorophyll has left the cell. And allow chlorophyll to leave. And this is also, you can see the reaction, to see the starch iodine reaction. To see the starch iodine reaction. Okay, so on the left is starch reacted. Iodine reacted. Reacted iodine, Lugol solution. But on the left, if you have no reaction, this is what it would look like. So we have no reaction here. Okay. And I'll explain the chromatography and the how do you pronounce this word? The spectrophotometry in the next video.